So I come from a very different background than Joel's. I was trained as a biochemist. My first project involved the mammalian brain, which I think we'll all agree is an extraordinary evolved structure of great complexity. What a biochemist does with the mammalian brain is you put it in a blender. And you fractionate it into its individual components, and in this case, the goal of trying to find one or two explanatory variables for a biological process. And I think we all understand now really the inadequacy of that approach, and we heard that very much from Joel, the sense instead that what we have to do is think of biology as a large set of interacting components. But it remains true as the technology for the collection of data has gotten ever better, that our ability to extract informative and actionable information from that has really lagged far behind. And it was really a nice setup from Joel's talk here to mine is that he talked very much about an analytical approach, which in this graph, which I took from James Brazzi, is very much on the left-hand side. That is to say, it's a discovery-based tool that relies entirely on unbiased approaches towards big data. In most scientific disciplines, there's actually a completely different and complementary set of approaches that really relies on simulation data. And in the biological sciences, the availability of large-scale genomic data has led to the relatively rapid development of methodologies for data analysis, first from genomics, and then we heard from complex sort of personal and clinical phenotypes. But on contrast, in the right-hand side of this, the sort of capacity to do multi-scale simulation has lagged seriously behind. And in fact, it's a largely underdeveloped side of biological discovery, and that's what I want to talk about to you today. And I want to start off with really an incredibly simple problem, actually, which is sort of illustrated here in this movie that is now playing. And this is just a set of cells. These are human tumor cells. We'd engineered them so we could figure out when they were about to die. They turned from blue to yellow as a consequence of their expressing an activity reporter. And over on the right, you can see the activity here digitized in these single-cell images. And what we find in these type of cells is that even though the cells have this sort of long period of ramping up, they go very quickly, independent of how long it takes them to make this eventual decision to live or die, a pretty consequential decision for cells. In our cells, in our bodies, for example, around 10 to the 9th cells will be born and will die today as a consequence of this regulated process. So regardless of how long it takes them to make that decision, once they make it, shown here in red with another one of these single-cell reporters, they make it unambiguously. So they transition from life to death. But what's striking is if you take a genetically homogeneous field of cells and you expose them to a uniform field of a ligand in this case, it's one of the TNF-like ligands that induces cell death directly by binding to its receptor. What you'll notice is they do so at different times. You could see that in my previous image. And some subset of them will survive. And if you take those surviving cells and you sort of replate them and grow them for a while and repeat the experiment again, what you'll notice is exactly the same phenomenon, namely that they will die at different times and the similar fraction of them will survive. Now, in cancer cells, they're sort of genetically unstable, so eventually you can take this experiment out after a couple of months to the point where you get frank genetic variation. But what you're looking at here is a variability in phenotype that is stable, intrinsic to the population, and we know arises from two types of underlying stochastic events, one of which is prior to the experiment, we call that intrinsic noise, and the other one happens subsequent to the experiment, we call that intrinsic noise. And the way these arise is, of course, in this conference we're thinking about biology as information technology, but in fact biology is chemistry. And what we really are large collections of complex dynamical systems that have very specific properties with respect to their underlying reactions. So in this particular case, we can go back and we can look at the graph that I showed you before where we're just looking at one of these enzymes downstream of the receptor. Here we're tracking it in single cells. And we simply have gone back post hoc and colored differently yellow and blue between the cells that lived and the cells that died. And I think you and I would agree if you simply looked at the activity of this initial step in the cell death process between here now, the distribution between blue and yellow, there's not an obvious difference between them. However, we know that this system doesn't simply consist of a bag of parts that, and I would say in contrast to what Joel described to you, that bringing in some prior information about the underlying chemistry of life can be informative. And in this particular case, what we have is the enzyme here. It's called caspase 8. It comes in and it's cleaving its substrate. They form a complex. We know that there is an initial encounter complex and a subsequent catalytic event. Turns out that there are about between 35 and 50,000 molecules per cell. So this is nicely approximated as a continuum dynamical system. And that means you can write a series of underlying equations that will describe it. So here's an incredibly simple case. We came in with this really simple idea of the underlying dynamical system. And we said, OK, here's our ligand. The death ligand is known as TRAIL. It binds to its receptor, which is known as DR4/5, turns on that enzyme whose activity we're measuring here in color. Now, the relationship between an enzyme and a stable substrate is going to be one of a simple derivative. So let's just go ahead and take a derivative of those trajectories that I showed you before, exactly the same data. And what you can see now here is 85 to 90% accurate discrimination between living and dying cells, simply based on the understanding that the relationship between the measured variable and the underlying biochemistry was one of a differential equation. So if you do that, you can go and you can build, in this simple case, you can build a landscape over the relevant parameterization of the problem, and you can very nicely discriminate between living and dying cells, here based on the properties, in this case the initial activity and the time scale of the reaction. You can put, and naturally live cells lie on the left of this, and dying cells lie on the right. And there's a little bit of error here, and we know that that arises from the intrinsic stochasticity of these chemical reactions, because we think of them as rates, but in fact they're truly probabilities. Now, the simple concept that I want to analogize here in this depiction, leaving aside the particular biology, is of course this notion of some sort of a phase landscape. And it turns out that this very simple notion, and Joel touched on it before, that these kind of processes, either at a cellular level or at a human level, actually operate over a series of variables is extremely difficult to get at in the absence of quantitative data discovery. So as a consequence, if you were to go to a Cold Spring Harbor meeting on quantitative biology, of which there are many, there are going to be fierce arguments among the community. Does it matter how fast this thing got going, or does it matter how strong it was? And what my little landscape here shows you, well, of course, it depends on the ratio of those two variables. And the analogy in the case of water is what's important for water freezing. Is it pressure or is it temperature? I think oceanographers think it's temperature. Glaciologists think it's pressure. To my knowledge, they don't have meetings where they have successive sessions, each of them arguing with the other about which one should be funded by the federal government. So you can actually then take these very simple dynamical ideas, and you can then go back, in this particular case, to a set of therapeutic antibodies which had been developed to agonize this pathway with the aim of killing cancer cells. So this is going to be a novel anti-cancer mechanism. It went through what we now call, in my lab, the classic ASCO cycle, American Society of Clinical Oncologists, where a new drug concept comes in the first year. There's a lot of interest. Second year it comes in, and it is in fact going to cure all disease, and all other discovery processes can be abandoned. Stock prices go up a couple of $1 billion often on those kind of discoveries. Go 3 or 4 years down the road, and pretty much they've hit clinical trials, and they're in the process of substantially underperforming, and eventually they become a failed therapeutic class. And we almost never go back and ask ourselves, why did that happen? In the cases where it's been looked at, from a sort of fairly general level, it turns out that often the underlying biological hypothesis was incorrect. So that the molecule, particularly in modern pharmaceutical development, the idea of building a selective agent to hit the target that you wanted, to getting that into the patient in an adequate concentration is well worked out. You do that, but it simply doesn't achieve the clinical endpoint. Here was a rare example where we went back. This is looking at Genentech's ApoMab antibody, which we resynthesized, and we simply went back and looked at it, and we found it bound to the target with very high affinity, nanomolar affinity. It's a therapeutic antibody in this case. In fact, you can see my little death landscape here. They're all living over in blue, didn't kill any cells. We could actually combine this with various proposed combination therapies. And what we find is this is a molecule which, binding very well, is simply not able to agonize the receptor. So a simple example of something that we would see all the way through an engineering discipline, namely of root cause analysis. When Boeing's 787s had a problem with their batteries. You'll recall they grounded them briefly, went out and tried to figure out what it was, fixed them, and then brought them back into the air. If that had been pharmaceutical development, those planes would have been taken to Washington. They would have been plowed into the Puget Sound. All of the relevant executives would have gone to another company so that they could start over again. And we wouldn't have either fixed the problem or learned the nature of the underlying flaw. So I want to leave you with the thought that one of the problems we have in biological sciences is taking this anecdotal, and you heard this from, as I say, from Joel in a data-driven, and I'm now telling you in a chemistry-driven way, of taking the complexity of interacting components and understanding their dynamical properties and then pushing that forward into a better understanding of how it is that those function in disease, and very critically for my own program, how it is you could go about drugging them for therapeutic purpose. Now, having adequate models of these processes relies, of course, on having good data. And the problem we have with data in biomedical science is, for the most part, we lose it all. So publication is largely a process of knowledge destruction today. And it is not the fault of the journals, it is the fault of the scientists. And it's really unique to genomics and some of the areas that Joel talked about, where the data is actually put out into the public domain contemporaneous with the paper. We estimate that 95% of all the data collected in translational, preclinical and translational biomedicine is in fact lost, and you can't find it. So go to a paper that you might find in an exciting review. We went to one from Mike Elowitz recently, which looked at the dynamical properties of transcription factors, an entirely new discovery, and you cannot find a single piece of underlying raw data from any of 35 publications in the top 5 journals. It simply is not stored and not recorded. So we've been fortunate as part of an NIH project called LINKS to actually work out how to liberate this kind of underlying data. And there's a website that I can guide anyone who's interested to. The hard thing is not actually to dump your data in the public domain. It's to dump it into the public domain in an intelligible way. So if you were at all interested in the phenomena that I showed you before, you could go to the paper and you could find something, but you can then, we now build a landing site for every paper, and from that you can go to every single piece of underlying data and you can pull out all of the dynamical trajectories here, either in a processed little picture or as raw underlying trajectories. And we've now had many examples, as genome scientists do, of people coming and looking at it and saying, hey, you know, you missed this feature altogether. Can you reanalyze it? And we say, we could, but we don't need to reanalyze it. You can reanalyze it and come back to us if you need some help. So why don't we do it routinely? The thing we've learned in doing this over the last 3 or 4 years is it's about 20% of our total effort to take a piece of laboratory data, and it's going to get harder as we get closer to the clinic, to process that into a generally usable form. The incentive for doing so, pretty much zero. Why do we do it in this case? Because we're being paid to by the LINKS project. But in general, we have not created a system of incentives in which the liberation of data outside of genome science is seen as critical to preclinical science. Now, actual signaling systems, and this was mentioned by Paul, are a lot more complicated than I've alluded to in this very simple little example of trail binding to its receptor. It turns out that system's gratifyingly complicated as well. So when we want to describe these underlying systems, as you're all well aware, we draw a series of pictures. And this is one actually around the RAS cascade that's the focus of the DARPA Big Mechanism project. Now, if you ask people in my contemporaries mainly, what do we know about the system, they'll say, we pretty much know everything about it, because in this case we have high resolution structures. So we actually have atomic resolution structures of the majority of the components, and we have a pretty good individual description of their biochemical activities. But what's missing is that we have very, very little understanding of the dynamical relationship among the components, and the fact that the same gene product works completely differently in different cellular settings, the contextualization that Joel talked about. So the thing that and there are also features, I should say, about this that we don't really know how to understand at all, and that is that if you think of a pathway that we draw here, it goes sort of A goes to B, and B goes to C, and C goes to D. But that's not how chemistry works, and it's been best understood in the folding problem here. And that is to say, we don't have single sequential reactions. We have massive concurrency of reactions. So, and this in the folding case is the only place it's been worked out for simple programs. We know that, in fact, multiple reaction channels are populated at the same time, and the relative balance between them can alter with fairly subtle differences in the underlying environment or in the protein itself. So as a first step in actually making simulation kind of actionable biology be feasible, we need to create a sort of tool system. And when I was at MIT for many years as a practicing biologist, practicing biochemist, grinding up wonderful organs and turning them into pink soup, I assumed that what we would do is we would just go to the chemical engineers, right? Because those guys are in charge of building reaction networks. And then we thought we would go to computer scientists, and that's, for those of you who know the now quite famous paper on building a radio, biologists, how they would fix a radio. And in each case, it's turned out that those analogies were inspirational, but so weak that they weren't really actionable. So biological systems just don't work like chemical plants. We just are not the same as electronic circuits. So it's been necessary to come back and create a new set of tools that will allow us to do this in a more effective way. We thought initially what we would largely do is build mathematical models. Over time, what we've learned from that is that it's simply too detailed and too error-prone. So what we're now focusing on in DARPA Big Mechanism is the construction and automatic validation of programming languages. And Paul mentioned a few of the underlying concepts there. And these programming languages are actually, in our particular version of it, they're just Python languages. They're domain-specific Python languages. So when we want to describe a biological system, we try to take all of the underlying features that people believe are correct or are uncertain about, and we try to instantiate them computationally, and then we try to test them against data. At any time, we don't have one model, one extant model. We have an entire family of alternative hypotheses. And one thing, if you actually formalize this, is you quickly realize, relative to my discussion earlier about Cold Spring Harbor meetings, is people are arguing fiercely about things about which you can pretty much show the data are equiprobable. We really don't know. And they also are very confident about things about which we could say the data tell us virtually nothing. So we have a very poor understanding of the subtlety of underlying data. One of the valuable things about beginning to push biology in this kind of computational framework, particularly dynamical systems, is it begins to introduce some abstraction layers. And all engineered systems, as you're well aware, require various levels of abstraction in order to make them intelligible. In biology, we don't necessarily know what those abstractions are. Here we can operationally, we have a high-level language, this programming language. It creates an equivalent set of things called rules-based languages that Paul meant. And then in the end of the day, we create mathematical equations. Ultimately, this is a mathematical equation-based approach. And that's exactly like high-level languages versus code versus machine code. So the same kind of analogy. And in fact, we put a whole bunch of these in. So we like to think in terms of working in terms of paragraph concepts, sentence concepts, and verb concepts, so that we have a kind of reusability for the underlying approach. Now, the result of doing this, first, is that there's a whole set of data that we can get at. In fact, we got at that little trail problem using this system. A PYSB model shown on the top, that's this domain-specific language, is actually human intelligible. You can go ahead and read it, and it says, the MAP kinase is operating on RAS. It has a phosphatase 2A opposing it. There's a double phosphorylation event, et cetera. So with a little training, you could all read it. It actually builds the underlying machine code. The chemical network reaction, here it's the instantiated network, which is not intelligible to you or me, and it's not intelligible to the person who created it in the first instance. It's simply too detailed to be a useful computational artifact. Importantly, in this system, pictures are derived from code. Pictures don't drive the development of the code. And that seems elementary, except in biology, figure 7, the conclusion of all studies is a picture of some sort. And for any piece of biology you're interested in, you can go find a bunch of pretty pictures. Those should be derived artifacts from a shareable computational infrastructure. And one of the first models that we're trying to build in this scheme that Paul mentioned is going to be a complete open source executable model of RAS biology. And we are building it by hand using the machine methods that Paul mentioned, although I'll have to tell him after this talk that the most recent machine reading didn't do quite as well as you might have hoped. The key was not to compare it to Harvard biologists. That was an easy metric. We compared it to some MIT biologists, and they did a lot better. So in any case, we're building this executable model to be free for the having, along with the underlying methods for doing it. As Joel mentioned, a lot of this really is, and these are just sort of the bits and pieces that are being worked into this. This is really an exercise not just in computer science, but also in social science. Now, one of the nice things about doing this is we begin to bring biology into what we already have in sort of engineering sciences. So in engineering science, we have blueprints. Well, biology, we have some blueprints. Those have been built for the last 30 years. They just connect all the bits and pieces. We have pretty pictures. Those are important to sell airplanes. We call those models in biology, and the fancier your journal, I think this came from Nature, the fancier your picture. But I don't know how you could do anything from this other than inspect it and say it was nicely drawn. What you do have in the case of the pretty picture for a 787 that I showed you is you do have an underlying simulation model that gives you some confidence that the thing will fly. And in fact, that's exactly the piece that we're really focusing on in this big mechanism project, to use tools from computer science in the open domain to create these kinds of means of comparing quantitative data to underlying hypothesis. And one of the neat ideas is we begin to overcome the problem that we all have, is that currently, with the exception of some genomic science areas, what happened is I will interpret the literature and go into my brain, and I'll have my understanding probably in light of the experiments, and then I'll explain it to you in this seminar or to Paul. And inevitably, he's going to end up with an approximate representation, and he has no way of going back and saying, that guy sounded plausible, but I want to test his idea independently. If you can move this, the obvious point is, if you can move this into the world of code and associated liberated data, then you can go ahead and argue. These are a couple of scientists in my lab arguing. We argue here on GitHub through the creation and fusion of computational models. And then when we get to the little the black flags here, it's when we decide we have to go back to the bench and actually test something. So there's a lot more to building this infrastructure. I'm going to go in the interest of time to go ahead of this. There's a lot more to building this infrastructure. It's really the purpose of big mechanism. And there's probably a much better way of showing you the results than I'm showing you today, and that's part of a new project that Paul's running on communicating with computers, so maybe next time we'll have prettier pictures. But as a coda all of this, and sort of with this point about information science and biology, I do want to say there are a lot of underlying concepts in information science which we have found useful to move across into the biological arena. And I want to just close with 2 slides, and this has to do with thinking about how controllable biological systems really are. And the sort of shocking observation that was made, it doesn't pop up here, that was made a couple of years ago within this concept of Shannon information is that the transmission between the outside to the inside of a mammalian cell typically has an information content of not more than about 1 bit. That means that cells can barely sense whether a ligand is present or not present. And in fact, that is the cell-to-cell variability, to come back to the beginning of my talk, that I mentioned to you about. Here's a population of cells. We know they're genetically identical because we follow them in the microscope and we pull them out. So they're genetically identical. We expose them to a uniform field of ligand. 80% die, 20% don't. That sort of looks like a pretty bad decision-making capacity. Should all die or should all survive? And so within that context, what we think of in terms of informational capacity is really low transfer in the Shannon world. And of course, the analogy here is the ligand is hitting the cell on the outside. That's the signal transduction system. In the middle is all those bits and pieces that we're now trying to write up a dynamical systems for, and then there's some response, in this case, death. Now, the interesting sort of realization we came to in the last couple of years is that this kind of biological variability makes it much harder to ascribe a decision to an individual cell, because yeah, maybe it'll die or maybe it won't. But if we look at cells in terms of populations of cells, now we find that this variability actually makes them much more controllable. And so what that really manifests itself is as a very shallow dose response curve. So give a little bit of ligand, a few cells die. A little more ligand, a few more cells die. And if this decision was in the absence of noise, it would be absolutely discrete at a single concentration. So this simulation makes the point that, in fact, this variability that we see now uniformly through biology means that the channel capacity at a single cell level is relatively lower. It appears so in this formalism, but our controllability of populations of cells gets ever better as the noise is exploited more effectively by the underlying biological system. And that'll be particularly true if it is a case, such as I've shown you today, of an immune type of editing reaction. Why would that be important? Well, there are 2 types of decisions that we can distinguish in cells. And those are where the single cell has to make a decision on its own, and it's a continuous decision. And the one we've looked at is actually neutrophils chasing bacteria through a solution. In that case, the single cell can make incredibly accurate decisions. We think in each time scale we're making a 5 to 6 bit decision. On the other time scale are these Or on the other hand, there are these binary decisions, live or die. If every cell in our body lived or died on exactly the same concentration scheme without exploiting noise, then in fact, we would have no controllability over complex tissues. And we think those are cases in which cells actually go and exploit the underlying chemical stochastic kinetics to achieve more effective decision making at the population level. So conclusions then, I think there's a lot of informal reasoning in my field about multi-component biological systems, exactly how I was trained. You purify the key protein, and then you reason about it in your head. You make no assumptions. One of the biggest problems I have with my colleagues who don't do this is they'll say, well, what I don't like about your RAS model, the one you're building with DARPA, is it has all those assumptions in it, right? Because when you build a numerical artifact, you enumerate your assumptions. They say, I don't make any assumptions about how this works. I just use the data. And you think, but in fact, in your mind, of course, this is sort of this infinitude of undeclared assumption. Model-based analysis, I think, typically shows that data is much richer than we think. For a bunch of empiricists, we now spend 50% of our time on data collection. On data analysis. If you look at the NIH budget, it currently spends about 6% of the total budget on data analysis, and that hasn't substantially grown in the last 10 years. These executable models that I gave you a very simple illustration of, I think they're actually one of the things they do is they dramatically improve experimental design. We think they're the natural counterpoint to the data-driven discovery that Joel described and will be essential, particularly when you want to get at therapeutics. And that many of the concepts that I showed you today have really not found a receptive audience in the halls of the National Institutes of Health, but in fact, we're very grateful to Paul, DARPA, and his colleagues for allowing them to explore in the mechanism here. So this work, of course, wasn't done by me, but the people in my lab who are named here. Thank you.